Hello everyone, I am Raphael Mourad, Associate Professor at the University of Toulouse in France. And today I will make a training on convolutional neural networks for genomics. So it's a bit more basic than the previous training that I've done for large language models for genomics. Here we'll, because it's more basic, we'll dive in more into the architecture of models we'll play by adding different layers to the models. So it will be interesting in a way that it will be a bit simpler. It's based on a previous tutorial that I have made a few years ago, and with the success of the large language on the tutorial on the large language models that I've done recently, I decided to revive this old tutorial on CNN for genomics. So here's to access to this tutorial, you just go to my GitHub repo and you go to CNN for genomics training here. So it will be a bit a smaller tutorial. There will be only two notebooks. The first notebook is about preparing the data, preprocessing the data and preparing the data. In particular, ChIP-seq and DNase-seq data. And that we'll do now. So we'll do this tutorial for now. So let's start. Let's go. Here we have the Google Colab. So what we need to do first is to clone the repo inside the Google Colab notebook. So let's do it. Then we'll so this just to say, so this repo is in R. We're not using Python because it's more convenient to work with R. For ChIP-seq data, there are many R libraries that are very convenient, such as genomic ranges, and we can easily extract also the sequences from the human genome. So because it's more convenient to work with R, it's a notebook working with R. So the good thing with Google Colab, you can use both R or you can choose R or Python. So how to do it is here. Instead of Python, here I used R. And I'm using a CPU because I don't need a CPU, okay? So here is, I'm gonna set the working directory of the file that I just downloaded, which is CNN for genomic training. And now we'll need to install, so by default, unfortunately, many R libraries are missing. So this is what is a little less convenient with Google Colab with R, is you have to install many R libraries, and it takes time sometimes. It can take time. So we have to wait here. What we are installing is we are installing the Human Genome Assembly HD19, and also it's a masked genome assembly, the mask. So it will take some time because the file is quite big. And at the same time, also, unfortunately, there are many dependencies with other R libraries. So not only we will download BS Genome R package, but we'll also download many R packages here that are listed here. So it will take around 10 minutes to download all of that. So let's download and install. I will talk a bit more about this tutorial and about the data. Let's talk more about the data. So the data are available in the folder data. And there is a folder that is called narrow-peak, which is what you get when you do, for instance, a ChIP-seq experiment. So ChIP-seq is chromatin immunoprecipitation sequencing. So for instance, imagine that, so I took ChIP-seq data from the ENCODE project in the GM 12878 cell line. And when you use, for instance, how does it work? You do the experiment, then you have FASTQ files with the reads, and then you map the reads, for instance, using BWA. I mean, that's what I did. I used BWA. And then I used Max for calling ChIP-seq peaks, meaning to identify where CTCF binds to the DNA sequence of the human genome. So at the end, with Max, you have a file that is called a narrow peak. So let's click on it and let's look at the file while the R packages are getting installed. All right, so here is what you get when you use Max as an output, the narrow peak. So it will provide you I mean, on the top, you'll get the peaks with the highest signal. So here you have a peak at the chromosome 14 of the human genome, HG19. The peak starts from disposition to disposition. It has a signal intensity of 549, and that's it. And then you have many other peaks. So here is a list of other peaks that we identified in the genome. Okay. So this is where CTCF was measured to bind significantly. And what we'll do here is we'll try to build a model that will take a DNA sequence and predict whether this DNA sequence binds to CTCF or not, for instance. So it's said here. So the idea here is to make a model for regulatory genomics. And for that, we want to predict from a DNA sequence whether it binds to CTCF or not. So this is when we use CTCF ChIP-seq. But if we use, for instance, ATAC-seq data, so whether we have a peak of ATAC-seq or not, so we will make a model that will predict, given a DNA sequence, whether there is ATAC-seq signal or not. So and there is a schema. I made a small schema here. You take a DNA sequence here, and then you'll predict through convolutional layers whether your sequence is positive, for instance, meaning that the DNA sequence binds CTCF, for instance, or it's negative, it does not bind CTCF. And it can work with CTCF, ChIP-seq, ATAC-seq, H3K4me3 ChIP-seq with many types of data. Okay, and so it depends on the data you give for training the model. And I provided many data, so CTCF ChIP-seq, DNase-seq. So I said ATAC-seq, but it was DNase-seq. I meant what I meant, which is the same. So basically, when you have a peak of DNase, it means that chromatin is open. So it assesses the openness of chromatin. H3K4me3 is a histone mark of promoter activity. JUND is a transcript factor that binds to DNA. P300 also is a transcript factor that binds to DNA. And POL2 is a polymerase II that is required for transcription of genes. Okay. So let's see if it got installed. No, it's still taking some time to install. All right. So let's wait a bit. Unfortunately, it takes some time for the installation. Okay, yeah, it's a bit slow. That's why R is not that very convenient, because for Python, many packages are available for Python. So it's quite quick to run Python notebooks in Google Colab. But for R, it's a bit less convenient. It's a little less convenient. Then after we'll have the GKM SVM R package to install, which is quite quick. Also, there are many dependencies. And then once all these R packages are installed, we'll be able to run the So just to say, so if you want to have an idea about what is a ChIP-seq peak, you can see, let's look, we can see online, for instance, here on this image. And here you can see that along a loci on a chromosome, you have a peak. So here there is a peak of CTCF, another peak, and a fourth peak. So here you can see that there are four peaks of the CTCF. Okay. And when you use a max, you identify those peaks that are significant, and then you can extract the underlying DNA sequence. Also, other thing that is interesting to see is that why can we predict whether a protein will bind to the DNA? We can predict because we know that for many proteins that bind to DNA, there is a DNA motif that the protein is recognizing. So for instance, for CTCF, we know that this protein often it binds to a given DNA motif. So this motif here actually it's a weighted motif, which was actually identified from ChIP-seq. So actually, the CTCF protein can recognize this motif on DNA. That's how the protein can bind to DNA. All right, let's go. So GKM SVM was installed. Also, BSGeom for the HG19 genome assembly was also installed. We can restart the notebook. So let's load the libraries now. Okay, so this was quite quick. Now let's define some parameters. So we will here what we will do is we'll make a binary classifier to distinguish DNA sequences binding to CF with DNA sequences not binding to CTCF. So we select the experiment CTCF, but we can also select other experiments like DNase or H3K4me3, for instance. So basically, you have all the data are here, and you just have to select CTCF, DNase, GND, P300 as you want, okay? There are four DNA letters in the DNA, so ACGT. We'll select only 4,000 peaks so that so that we have data for training that is not too big. And also we'll resize the peaks because the peaks can be large or less large, and we'll select a size. We'll resize the peaks with a size of 201 base pair, so that all of the DNA sequences are the same size. So here, sorry. So I'm clicking here, then here I'm defining the different file names. So we'll make two bed files, one bed file for the positive sequences, one negative file for the negative sequences, one bed file for the negative sequences, sorry. One FASTA file for the positive sequences and one FASTA file for the negative sequences. Here is where I define the human genome from which I want to extract the DNA sequences corresponding to the peaks. Also here, just to say that I only kept chromosomes from 0 to 22 and the chromosome X. I didn't keep any scaffold or chromosome that are not well built. Then here I'm loading the narrow peak file. So this is a table as I showed you before. The first peak was found in chromosome 14. It starts at this position, it ends at this position, it has this signal. Here I'm converting this data frame into a genomic range. Genomic range is object. And then it's a very convenient R library, and I can do many things. And here what I do is I resize the peaks. So here is originally I have so we have 45,000 peaks. I make them, I convert the peaks into a geometric ranges object, and then I resize the peaks to 201 base pair. All right, here I select the K best peak, so I wanted to have 4,000 peaks. I'm only selecting the best 4,000 peaks, meaning the peaks with the highest signal. All right, I save the file as a bed file. Actually, it's a positive bed file here. So it's just a bed file with a coordinate of the best 4,000 peaks. And here what I'm gonna do, it's an important part is, okay, we know that we have peaks corresponding to the DNA sequences binding CTCF. We also want negative sequences, meaning sequences not binding to CTCF. The problem is if you draw negative sequences randomly in the genome, you will not get at the end the best control sequences. One good way is because you'll have biases, you might have confounding factors that are different between your positive sequences and your negative sequences. So the best way is to make control sequences that are very similar, the most similar to the positive sequences. So what you want to do is you want to draw in the genome negative sequences, meaning sequences not binding to CTCF, that are kind of similar to positive sequences, meaning sequences that bind to CTCF. So for that, we'll use a function from the package GKM-SVM that will draw negative sequences that have a GC content and repeat distribution that is similar to the experimental to the positive peaks, to the experimental peaks. So let's do it. And with this function, so it will take a few minutes. And with this function, it will generate automatically the bed file of the negative sequences, the FASTA file of the positive sequences, and the FATA file of the negative sequences. So just to tell you, just to remind you, the bed files correspond to peak coordinates, and the FASTA files correspond to the underlying DNA sequences. And for deep learning at the end, we'll only use the FASTA files. All right, so it has been done. So we generated the control sequences. And then there is some code that I've done because there is a little bug inside the get_new_seq function. For some reason, it removes one base pair. So initially, my sequences were 201 base pair long. It shrank it to 200 base pair, and I have to make it again as 201 base pair long. So this is a code where I do that. So I'm uploading again the negative sequence bed file, adding one base pair, getting the corresponding DNA sequence, saving the FASTA file, and that's it. So at the end, the summary is the following. We have generated two FASTA files, one FASTA file for containing 4,000 positive sequences that are binding CF protein, and also around 4,000 negative sequences that are not binding CF protein. And with these two files, with these two sets of DNA sequences, we will be able to build deep learning models such as convolutional neural networks as binary classifier to distinguish between these two types of DNA sequences. So see you for the next training, for the next Google Colab for making the model.